So, today I'll be talking about bacteria to eukaryote horizontal gene transfer and a little bit about my experiences doing this independent research project. First, I'll show you what horizontal gene transfer is, and then I'll talk a little bit more about my experiences doing this project. So, we start off with one species of bacteria. And then, in its physical proximity, we have another species of bacteria. Some of the DNA inside the first species gets released into the surroundings when the bacteria bursts open, and that piece of DNA is taken up by the other species and becomes part of its DNA. It just involves a strand of double-stranded DNA, and while the cell is replicating, a piece of that foreign DNA from the different species goes inside the other species' DNA. Now over here it says fungal DNA because in my case, instead of a different bacterial species, we had a fungus species. So it all began when I was working actually on a different project for my lab. It involved creating like a sort of a clustering algorithm, a program that clusters together similar proteins. But I was working on that project, and in the process of that, I noticed that one of the proteins was found in almost every bacteria, like a lot of bacteria. But it was found in only one eukaryote or multicellular organism, and that struck me as odd. So I was like, why is this the case? Was this like in the common ancestor of all life and then it got lost in all eukaryotes except one species of fungus? Probably not. So that was already suspicious to me. So what I did was, there's this program called BLAST. When you put a protein into it, it gives you all the different proteins that are like it. And when I put the protein, the eukaryote protein from a species of fungus into the program, it showed that it was most closely related to a bunch of specific bacterial species that are actually, so that fungus is found in the intestines of cows, sheep, giraffes, this family of animals. And that bacteria, the one it's most closely similar to, that is also found in that exact same place. So that is actually how I figured out that this was a horizontal gene transfer. The best part of the process was actually the whole process of discovery, because that actually made me feel like a scientist, like an actual scientist, like, oh wow, like I discovered something new. I would say the whole discovery process, the whole, oh wait, is this really like what I think it is? And then doing some tests, I think that was probably like the best part of it. My advice to other undergraduates would probably be, don't be afraid to pursue your own project. Don't be afraid, don't underestimate yourself. If you notice something suspicious while doing work, first of all, be alert, and second of all, don't be afraid, don't underestimate yourself, don't tell yourself that, oh, this is probably not anything important, I'm probably misjudging myself, probably misinterpreting things. No, I would say that, believe in yourself and go and pursue whatever it is that you think is worth pursuing.